I want you to picture something for a second.
Okay.
Imagine you're standing at the top of this really bumpy, uneven hill, and you drop a marble.
Okay.
And as that marble rolls down, it starts hitting, little ridges, and it bounces into small valleys, branching off into all these different paths until it finally comes to a complete stop at the bottom.
Yeah, it is such a classic image. I mean, in developmental biology, we refer to that as Waddington's landscape.
Right.
It was proposed decades ago to help visualize exactly how a cell makes decisions.
Yeah, and instead of a marble, we're talking about a single generic stem cell.
Exactly.
As it rolls down this metaphorical landscape, it's losing its limitless potential, right? It's figuring out its final identity. Like, will it slide into the valley that makes it a red blood cell?
Mm-hmm.
Or maybe it bounces a different way and becomes a skin cell or a neuron.
Right. And the holy grail for biologists has really always been trying to map out that exact landscape. We want to understand the invisible forces that push the marble down one specific valley instead of another, because if we know that, we understand how life builds itself from scratch.
Which brings us to the mission for today's deep dive. We are looking at this incredible 2026 paper published in the journal Cell.
Yeah, a massive paper.
It really is. It's titled RegVelo, Gene Regulatory Informed Dynamics of Single Cells by Wang and a pretty large team of researchers.
Yeah.
And our goal today is to unpack how this team has finally built like a mathematical bridge between the speed at which a cell changes and the internal software that actually programs that change.
To fully grasp why RegVelo is sending such shockwaves through computational biology, we really have to look at the massive blind spot that researchers were dealing with before this paper even came out.
Right.
Historically, we had two incredibly powerful but completely isolated tools for studying cells.
Let's lay those out for everyone listening. The first one is a concept called RNA velocity.
Yes.
And I think of RNA velocity as like a cellular speedometer.
That's a great way to put it.
It doesn't just tell you what a cell is doing right now. It measures the speed and the direction of the cell's development to predict what it's going to do next.
Right. And the second tool is what we call a gene regulatory network, or a GRN.
Okay.
Think of the GRN as the circuitry.
Yeah.
It is the genetic software map that dictates, which genes are turning on and which genes are turning off to actually drive the cell's development.
So we have a speedometer and we have a wiring diagram.
Exactly.
But here is the crazy part.
Yeah.
Before RegVelo, biologists had absolutely no way to connect the two.
None.
It's like, imagine sitting in a car with a speedometer that tells you you're accelerating, and you're holding a schematic of the engine's electrical wiring in your hands, but you have no mechanical way to see how pressing the gas pedal actually changes the speed.
Yeah, it was a profound conceptual disconnect. We could watch the cells move along their developmental paths, and we could draw these static maps of gene networks, but we just couldn't link them mechanically. We had literally no mathematical way to prove how the wiring was driving the motion.
So to understand how they finally solved this, I feel like we really need to lift the hood and look at how that speedometer, the RNA velocity, actually works.
Good idea.
Because from what the sources show, the old way we calculated it was, frankly, a little broken.
Oh, it was heavily flawed. But, the underlying biological mechanism of RNA velocity is actually quite elegant.
Okay, break that down for us.
It relies on the life cycle of RNA inside the cell. When a gene is activated, it doesn't instantly produce a perfect finished protein.
Right.
First, it transcribes this rough draft, which we call unspliced RNA.
It's the raw, newly minted instruction manual.
Exactly.
It still has all the biological junk in it that hasn't been edited out yet.
Yes. And then the cell's machinery comes in, cuts out the unneeded parts, and processes it into what we call spliced RNA.
That was okay.
That is the mature, ready-to-use instruction.
This makes perfect sense when you look at how scientists actually use it to predict the future. Like, by looking at the ratio of raw, unspliced RNA to mature, spliced RNA, you can tell if a gene is ramping up or powering down.
Precisely.
It reminds me of walking into a kitchen.
Oh, I like this.
If you look on the counter and see raw ingredients, flour, sugar, a carton of eggs, and nothing is in the oven yet.
Right, you know what's coming.
Exactly. You can predict with near certainty that the chef is about to bake something. You are literally looking at the future state of the kitchen by just measuring the raw materials.
That ratio of raw to mature RNA allowed previous mathematical models like scRNA-seq to create these really beautiful vector fields. It could literally draw arrows showing exactly where a cell was heading.
Okay, but
But yeah, to make that math work, those older models had to rely on some incredibly restrictive and frankly unrealistic assumptions.
This is where the old models hit a massive wall. What exactly were they assuming? Because I know the math gets a little dense here.
Well, they assumed that every single gene in the cell operated completely independently of every other gene.
Independently?
Yeah. In mathematical terms, they used what's called decoupled one-dimensional ordinary differential equations, or ODEs for short.
Wait, I need to stop you there. They assumed genes operate in a vacuum.
Basically, yes.
That's like trying to understand city traffic by focusing a camera on one single car while completely ignoring the traffic light, the stop signs, and the hundreds of other drivers swerving around it.
It is a severe oversimplification, yes. But it was the only way they could get the equations to resolve at the time.
Wow.
Decoupled just means the equation for gene A simply doesn't care what gene B is doing. And furthermore, they assumed the speed at which that raw RNA was being produced, the transcription rate, was just a constant, unchanging background hum.
But biology is basically the exact opposite of a vacuum.
Exactly.
It's a highly interactive, super messy system. You have upstream genes acting like traffic cops, constantly turning downstream genes on and off.
And those upstream genes are called transcription factors. By excluding them, by ignoring the regulatory traffic cops that actually drive a cell's differentiation, those old RNA velocity models were fundamentally missing the underlying mechanism of Waddington's landscape.
They could see the marble rolling, but they had absolutely no idea why it was rolling.
Precisely.
Okay, so let's do a quick recap for you listening, make sure we're all on the same page.
Yeah, I do.
Bullet 1, we have Waddington's landscape, which is our mental image of how a cell decides its fate, rolling down a hill.
Yep.
Bullet 2, we have RNA velocity, our speedometer that predicts future cell states by looking at raw versus mature RNA in the kitchen.
Perfect.
And bullet 3, the old mathematical models completely ignore the genetic wiring, the GRNs, that connect all these changes.
Because they use those decoupled one-dimensional equations.
Right. So the big question for you, the listener, is how do we actually fuse this wiring map with the velocity prediction? How do we build a better model?
And that is exactly where the authors of this paper come in. They developed a framework called RegVelo.
RegVelo.
It stands for regulatory velocity. And rather than relying on those old decoupled equations we just talked about, RegVelo is built on deep generative modeling.
Okay.
It merges the RNA velocity equations and the gene regulatory networks into a single high-dimensional ordinary differential equation.
A high-dimensional equation.
Yeah.
Okay, let's translate that out of math speak for a second.
Sure. If the old models were thousands of isolated single-lane dirt roads, RegVelo is a mathematical superhighway system where every single road intersects.
Oh, I see.
Transcription is no longer modeled as a constant isolated event. It is now treated as a dynamic process that depends entirely on the regulations of other genes and the passage of time.
I found the mechanics of how they actually built this superhighway fascinating because RegVelo doesn't just guess how these genes connect out of thin air, right?
No, it can't.
It needs a starting point.
Yeah.
So the scientists feed the algorithm a starter map of the gene regulatory network.
Right, and they derive that starter map from other types of single-cell data. Often they use something called multi-omic data, which involves looking at the physical accessibility of the DNA itself to guess which genes might be able to talk to each other. We refer to these starting maps as epigenetic priors.
Epigenetic priors.
Yeah.
I love that concept.
Right, yeah.
It's like looking at the physical shape of the DNA, like seeing which chapters of the instruction manual are physically open and readable to guess the wiring diagram.
That's great analogy.
So they take that rough starter map and they feed it into a deep neural network.
And that neural network has to calculate something absolutely critical called latent time.
I was really hoping we'd get to latent time.
Oh, it's so cool.
Because when you scoop up a sample of cells to study them, you aren't capturing them all at the exact same moment in their life cycle.
No, it's a mixed bag.
Right. So latent time is like the algorithm calculating an internal biological clock for each individual cell, placing them in perfect chronological order along a timeline of development.
It is a crucial step. And alongside that internal clock, the neural network also generates a massive matrix of weights to represent the regulatory network.
A matrix of weights.
Yes. It is assigning a mathematical score to how different genes interact with each other.
This weight matrix is where this concept really clicked for me when I was reading the paper.
Yeah.
Yeah. If the neural network observes the data and gives a connection a positive number, it means gene A actively turns on gene B.
Yes.
And if it gives it a negative number, gene A represses or shuts down gene B.
Exactly. And a 0 means those two genes simply don't interact at all.
Okay.
But here is the true stroke of genius in the RegVelo framework.
What's that?
It knows that biological starter maps, those epigenetic priors we talked about, are notoriously noisy and imperfect.
Yeah.
It does not blindly trust the prior information it was given.
Because in reality, a lot of those initial guesses about which genes talk to each other are just flat out wrong.
Exactly. Biological networks are governed by a principle called sparsity.
Sparsity. Let's define that.
It just means not every gene interacts with every other gene. Most only communicate with a highly specific select few.
Right.
So RegVelo's deep learning model uses the actual RNA velocity data to learn new edge weights. It essentially self-corrects.
That's wild.
Yeah. It fixes erroneous assumptions in the original map and discovers hidden connections that the researchers hadn't even mapped yet.
It's rebuilding the car's wiring diagram while the car is actively driving down the highway.
Yes.
That's incredible. So time for another quick recap.
Let's do it.
Bullet 1, RegVelo merges RNA velocity with those gene regulatory networks.
Yeah.
Bullet 2, it uses complex coupled math, those high-dimensional ODEs, instead of isolated equations.
Right.
And bullet 3, it refines imperfect prior knowledge using deep learning, keeping sparsity in mind.
Perfectly summarized.
So, for you listening, think about this question. If we have this highly accurate, mathematically coupled virtual model, what happens if we start tinkering with it?
And this is where RegVelo transitions from being just an observational tool to being a full-on experimental playground.
Oh, this part is so cool.
The researchers connected RegVelo to an analytical tool called CellRank-2.
CellRank-2.
And this combination allows scientists to perform what are known as in silico perturbations.
In silico, meaning entirely inside the computer.
Exactly. They have essentially constructed a fully actionable virtual cell.
I'm trying to picture what this looks like in practice. Because if I'm a researcher studying a specific organ, normally I'd spend, a year breeding mice?
Oh, easily.
Chemically altering their DNA and waiting to see what a specific gene mutation does.
Yeah.
But with this, am I literally just typing code to delete a gene and watching the virtual organ break?
That's exactly what you were doing. You go into the matrix, you digitally erase a gene and all of its downstream targets, you run a forward simulation, and you watch the mathematical consequences ripple through the cell's fate.
Wow.
It allows for counterfactual inference, asking the model, what would happen if this specific traffic cop disappeared?
So to prove this wasn't just a fancy math trick, the team obviously had to test it on some highly researched biological systems.
Naturally.
And I know they started with mouse pancreatic development.
Yeah, the pancreas is a great testing ground because we already know a lot about how its different cells develop, like the insulin-producing beta cells and the ductal cells.
Right.
When they fed the raw pancreatic data into RegVelo, the model independently identified the known driver genes.
Which is a huge first step.
Yeah.
And then, when they did a virtual knockout of a gene called E2F1,
Yes.
the simulation instantly showed a massive disruption in the cell cycle of the ductal cells.
Exactly.
And then when they digitally knocked out a gene called RFS6, the virtual cells suddenly failed to differentiate into beta cells.
It perfectly mirrored decades of hard-fought, real-world biological experiments just by crunching the data.
But an even more striking example from the paper involves human hematopoiesis.
The formation of blood cells. Yes.
This system relies on a classic, textbook example of gene regulation known as a toggle switch.
This is one of my favorite parts of the paper.
Let's break it down. It's the tug-of-war between GATA1 and SPI1.
Yes. And SPI1 is also commonly known as PU.1, just for anyone who might know it by that name.
Okay, so GATA1 versus SPI1.
When a human stem cell is preparing to become a blood cell, it faces a critical fork in Waddington's landscape.
Right.
It can slide down one valley to become an erythroid cell, which is a red blood cell that carries oxygen.
Okay.
Or it can slide down a completely different valley to become a monocyte, which is a white blood cell that fights off infections.
And the thing deciding which valley the cell falls into is a literal biological tug-of-war between those two transcription factors.
Exactly. GATA1 and SPI1 mutually repress each other.
So if GATA1 gets the upper hand, it physically blocks SPI1 from functioning, and the cell becomes a red blood cell.
Yes. And if SBI1 wins the tug-of-war, it shuts down GATA1, and the cell becomes a monocyte. What is so impressive is that RegVelo captured this toggle switch perfectly. The researchers went into the virtual model, computationally deleted the GATA1 network, and they watched the mathematical fate probabilities instantly shift entirely toward the monocyte lineage.
It's a perfect digital mirror of biological reality.
It really is.
And it's worth noting that the older decoupled RNA velocity models totally failed at this.
Miserably. Because they couldn't see the regulatory traffic cops, they couldn't predict the tug-of-war.
Even when previous models were fed extra highly detailed metabolic data, they still couldn't accurately predict these driver genes because they fundamentally lacked the regulatory context.
That's the power of coupling the math.
Okay, time for recap number 3.
Go for it.
Bullet 1. RegVelo acts as a virtual cell, allowing in silico or computer-simulated gene knockouts.
Yep.
Bullet 2. It successfully mapped pancreatic cell drivers like E2F1 and RFY6.
Correct.
And bullet 3, it perfectly simulated the GATA1 versus SPA1 toggle switch in human blood cells.
Awesome summary.
But, if you're hearing this and thinking, okay, that's really cool, the computer built a nice virtual cell that confirms things we already discovered years ago in textbooks.
Right, confirming the knowns.
Yeah. But does this algorithm actually have the power to discover something completely new?
And that is exactly what the researchers wanted to prove next. Because validating known biology is one thing. Predicting unknown biology is the ultimate test of any computational model.
So to do this, the researchers chose one of the most complex, chaotic systems in developmental biology.
The zebrafish cranial neural crest.
The neural crest. I mean, this is Waddington's landscape on extreme fast forward, isn't it?
Oh, it truly is. These are a temporary group of cells in a developing embryo that migrate incredibly fast and differentiate into a wildly diverse array of tissues.
Like what?
They turn into the pigment cells that give the fish its stripes, the cartilage in its face, peripheral neurons, glia. It is a very noisy, highly dynamic system.
So to give the algorithm the best possible chance at mapping this chaos, the researchers didn't just use standard sequencing.
No, they couldn't.
They threw the absolute state-of-the-art at it, generating a massive multi-omic data stack.
They needed extremely high-fidelity input, so they used a technique called full-length SMART-C3 to get incredibly high-resolution RNA data.
SMART-C3.
Yes. But more importantly, they measured that RNA expression while simultaneously measuring the chromatin accessibility.
Meaning how physically open the DNA was.
Exactly, in the exact same individual cells.
So they were essentially reading the text of the instruction manual while simultaneously checking the spine of the book to see which pages naturally fell open.
That is a brilliant way to picture it, and it gave RegVelo an unbelievably rich starting map.
So they fed all of this high-resolution data into the framework.
Right. And RegVelo's algorithms crunched those high-dimensional equations, calculated the latent time, self-corrected the network weights.
Using sparsity, like we talked about.
Yes. And it output a prediction. It pointed to specific, very early transcription factors as the master switches for pigment cell development.
Specifically, it flagged 2 genes, TFEC and ELF1.
Now, scientists knew these genes were involved in development somewhere.
Right.
But their role as the primary earliest drivers, dictating the pigment lineage, was heavily underappreciated. RegVelo basically looked at the math and said, these 2 genes are the most critical traffic cops at the very top of the hill.
And this is honestly my favorite part of the entire paper.
Oh, the validation.
Yes. The researchers didn't just publish their neat little computer simulation and walk away.
No, they didn't.
They took RegVelo's mathematical prediction and marched straight into the physical wet lab.
They performed what we call in vivo knockout validation.
Right.
They tested it in living animals.
They took actual living zebrafish embryos.
Yes.
They used CRISPR-Cas9 genetic scissors to physically snip the TFEC and ELF1 genes right out of the DNA.
And then they let the embryos grow and used advanced single-cell sequencing, specifically perturb-seq, to measure exactly what happened to those physical cells.
And the results were undeniable.
The physical biological knockout perfectly validated the digital mathematical simulation. When TFEC and ELF1 were removed from the living fish, the pigment lineage specification completely broke down. The cells entirely failed to differentiate into pigment cells, matching RegVelo's virtual prediction flawlessly.
The algorithm nailed it. The virtual model was so tightly coupled to reality that it predicted a physical outcome in a living organism.
And the statistics back up just how significant a leap this is. When the team measured how accurately RegVelo predicted the downstream effects of knocking out these individual transcription factors, RegVelo achieved a mean Spearman correlation of 0.52.
Okay, I know a Spearman correlation of 0.52 sounds like deep statistical jargon.
It is a bit dense, yes.
But let's put that number in context for the listener. Compared to the older decoupled RNA velocity methods we talked about at the beginning of the show, how much better is that 0.52?
It is nearly double the accuracy.
Double.
Yes. By connecting the gene regulatory network to the velocity dynamics, they doubled their ability to predict complex biological reality.
That is huge. So let's do our final mini recap. Bullet 1. RegVelo was tested on the extremely complex zebrafish cranial neural crest.
Yep.
Bullet 2. It predicted TFEC and ELF1 as the crucial drivers for pigment cells.
Right.
And bullet 3. CRISPR-Cas9 knockouts in live embryos proved the computer model was right.
Spot on.
So the question to ask yourself now is, what does this actually mean for the future of how we study diseases and developmental biology?
Well, it means we are moving from simply drawing lines on a graph and observing a car drive by to actually understanding how the engine turns the wheels.
It represents a fundamental paradigm shift in computational biology.
It does. We are transitioning from an observational phase into a causal phase.
Right.
By honoring the fact that biology is highly coupled and interactive, RegVelo provides an interpretable, actionable framework. We can now test hypotheses in a computer simulation with a level of mechanical confidence that simply did not exist before this paper.
And that leads to a thought I really want to leave you with as we wrapped up today's deep dive.
Yeah.
Think about the implications of this for medicine. We just spent a lot of time talking about predicting how a healthy stem cell becomes a blood cell or how it becomes a pigment cell in a fish.
Mm-hmm.
But Waddington's landscape isn't just about healthy development. Cancer is essentially a problem of cell fate and gene regulation gone horribly, tragically wrong.
Exactly. It is a mutated landscape where the marble gets trapped in a valley it was never supposed to enter, and it just keeps dividing.
So if we now have the computational power to accurately simulate a virtual cell and predict exactly how altering its wiring changes its fate.
Where does that lead us?
Right. How far are we from a world where a doctor biopsies your specific tumor, they sequence the RNA, they measure the DNA accessibility, and they upload your exact cancer cells into a framework like RegVelo?
They could create a completely personalized digital twin of your tumor.
Yes. And instead of testing highly toxic chemotherapy drugs on your actual body through months of agonizing trial and error,
the doctor performs in silico perturbations.
Exactly. They digitally simulate thousands of drug combinations in the computer, knocking out different transcription factors until they find the exact molecular mechanism that forces your specific cancer cells to stop growing.
Or forces them to differentiate into harmless tissue.
Yes. They solve the puzzle virtually before you ever have to swallow a single pill.
That is the ultimate promise of this technology. By finally linking the genetic software to the cellular dynamics, we aren't just observing biology anymore. We are learning how to actively rewrite it.
So the next time you picture that single marble rolling down the bumpy slopes of Waddington's landscape, bouncing between the ridges and valleys, remember that we are no longer just watching it roll.
No, we're not.
We finally have the tools to read the map, and maybe very soon we'll be the ones drawing the valleys.